Hi, I'm Mary McQuilkin, adjunct faculty for the University of Alaska Fairbanks Center for One Health Research. Today, I'm joined by laboratory team lead Jeff Doty of the CDC Arctic Investigations Program in Anchorage. This interview will be about the virus formerly known as Alaskapox.
I'm Jeff Doty. I'm the laboratory team lead at the CDC Arctic Investigations Program, located here in Anchorage, Alaska.
Can you explain how the virus was first discovered?
It was really interesting, actually. So there was a person in Fairbanks in 2015, July of 2015, and she presented to her doctor with what she thought was a spider bite on her shoulder. And it hadn't been healing up, and the clinician was taking a look at it. He saw some signs that he thought where it might indicate a viral infection. And so he decided to do due diligence and took some samples and sent them to the Alaska State Public Health Laboratory. And they did some initial PCR testing, and it showed that it was an orthopox virus. So then at that point, they shipped the samples to CDC Atlanta to the poxvirus and rabies branch, and they did some DNA sequencing and determined that it was a novel virus. And at the time, they didn't have a name for it. They were just calling it Alaska virus, I think. Part of the interesting story here is really looking at where the source of the virus came from. She lived in Fairbanks. She had no travel outside the United States, but she had a partner who was working in Azerbaijan in the oil fields, and he was going over there and spending time working and then coming back to Alaska, but his last trip hadn't been for about 6 months. And so that's really long time for an incubation period. Once someone gets infected with a virus, they develop, or the virus will amplify, and then they go through the course of infection. But 6 months is out of the range at all for known for these types of viruses. So it wasn't likely that the person may have been infected from the partner. However, the partner did bring back some materials from Azerbaijan, including like a leather jacket that the case really enjoyed wearing, a wooden box, and some other materials. And so the thought was, well, maybe the virus could have been attached to one of these items that were brought back from this person's trip to Azerbaijan. And so they did take some samples from those materials, and everything came back negative. While we don't really know about the host systems of a lot of orthopox viruses, when we do have good quality data about the host systems of these viruses, it generally goes back to small mammals, including like rodents and shrews. And so that was another point to really hit here in the initial investigation, was to try to see whether there was any evidence of a zoonotic source. And so there were a small number of animal samples collected from a nearby location, and nothing came back positive from that either. One big reason why there was this Azerbaijan connection and why people wanted to track this down fully is that when you take a look at the phylogenetic tree of orthopox viruses, where this falls out is kind of between the New World orthopox viruses and the Old World orthopox viruses. And just a couple of years prior, in 2013, there was another novel orthopox virus that had been detected in the former Soviet Union country of Georgia, which borders Azerbaijan. And it actually kind of falls out in a similar topology on the phylogenetic tree where it's close to this Alaska virus. So all of these pieces together were really leading people to think, well, maybe this wasn't something that was picked up in Alaska, and maybe something that might be associated with the partner's travels. The authors of the publication did determine that the most parsimonious or the most likely explanation is that it was a locally acquired infection. And so that was kind of like the final conclusion of the paper there.
For those who may not be familiar with phylogenetic trees, can you briefly explain what they're looking at and why it's significant that the Fairbanks cases are all grouped together separately from the Kenai case?
Basically, what these phylogenetic trees do is it takes all the sequence data and it looks how similar the different data is. So this is a string of A, G's, T's, and C's. And, it's a code of life, building blocks of life for all animals or for all life. So these trees basically take all the data from the sequences of the viruses and look at which specimens are similar to each other. And so you'll see the branching on the nodes of the tree. Different horizontal link branch indicates different levels of divergence. So longer horizontal branches mean those samples are more divergent, more different from the neighboring sequences, whereas very short horizontal branch links indicate that those samples are close together or very similar. And so when you look at the tree, you see the big clumping at the top of all the specimens coming from the Fairbanks area, and they're all grouping out together. And there is some substructure underneath there. And you can see some of them end with MR and SC and HS. That indicates whether they're animal source or human source. And all of the human and animal sequences together from the Fairbanks area are grouping out together in that top group. And down at the bottom in the Kenai Peninsula, you can see those two, both the human and animal sequence are there together. The most significant finding here related to this is that this shows that it's very likely that the source of this virus was on the Kenai Peninsula rather than coming from somewhere outside. And so the fact that the human sample and the animal sample from the Kenai are very similar, and they group out so far separately from all the Fairbanks samples. That's a very strong indication that this was a locally acquired infection on the Kenai Peninsula and not associated with any travel or movement of animals from the Fairbanks area.
Do you predict that this is a virus that might become more of a problem for human populations in the future?
5 years ago, I would have said no. I would have thought absolutely not. Right now, there's no evidence of human-to-human transmission of this virus, and then there's only 7 cases known to date. However, there have been a lot of changes recently. In fact, Mpox, if people are familiar with the Mpox spread and from 2022, if these viruses get into a target population that maybe isn't how they evolved, things could start changing as far as these transition dynamics. And so it's always a big unknown what's going to happen in the future and what those kind of special populations or risk groups may be.
You mentioned that genetically the virus is similar to a virus that was found in completely different parts of the world. Is it likely that these are ancient viruses that have been around since humans have been around, do you think? Or do you think these are newer emerging pathogens?
I do think that they have been around for a long time. So these are older viruses. The structure within the phylogenetic tree and the amount of genetic diversity within the group of the orthopox viruses in general is very robust. So there's a lot of different species, and the diversity within those groups is strong. So I think it's very likely that these viruses have been around a long time. This isn't a new virus or anything. And one thing, if you look at the distribution of the northern redback vole, it has this whole Arctic distribution. It's in the Arctic in both the New World and in the Old World. And when you think about that in relation to the larger phylogenetic tree that shows the position of all orthopox viruses and where Borealpox virus is in, where it is between the New World and Old World viruses, that goes a long way as far as explaining that this virus may have evolved in the Old World and then came across the Beringia area. That's really hypothetical at this point, but it goes back to this virus being similar to the virus over in far Western Asia.
How were the initial epidemiological investigations conducted in Fairbanks?
Sure. So the epi investigations were primarily conducted by the Alaska Division of Public Health. And so they would go interview the patients, ask them about timeline of symptom onset, activities that they were, partaking in the time, try to identify any maybe some high risk activities that might have made someone more likely to be infected. There have only been 7 cases ever reported so far, and 6 of those were from the Fairbanks area. So the initial case was in 2015, and then there were additional cases in 2020 through 2022. And all those cases were in the Fairbanks area. So when the 2020 case happened, it was kind of like an eye opener, say, okay, well, this 2015 case wasn't just a one off. We really need to dive in deeper here and a more robust effort to try to look for a zoonotic source of this virus. And so the Alaska Division of Public Health invited people from the CDC Atlanta, the Poxvirus and Rabies branch, which I was part of that team, up to Fairbanks to come do an investigation. So we ended up sampling animals at 15 different sites around Fairbanks. We ended up doing an investigation in 2020 and in 2021. And in total, approximately 385 animals were sampled during those efforts. We use different kinds of traps as far as catching these animals. So we'll use all live traps. So the animal is in there and captured alive. So the general styles we have are tomahawk live traps, which are kind of a cage style trap we use for generally catching squirrels and maybe slightly larger mammals rather than like a vole, but a small vole. We use a small trap called a Sherman live trap, which is also kind of like a metal cage, but it doesn't have any perforation, so you can't see the animal inside. And then we also use pitfall traps, which are basically a plastic cup that's buried in the ground. And these are designed to capture a shrew. So a shrew is just walking along, mind its own business, and falls in the hole and then cannot escape. And so we can just reach our hand in there and pull it out of the hole at that time. So really, these are the types of tools we had as far as try to collect samples from the animals. When you're in the field conducting these types of investigations, you just can't go out without proper personal protective equipment. And so we had to use biosafety level 3 precautions. So we had to wear powered respirators. We wore gowns over our clothes, definitely something over your face and eyes to prevent any kind of splashing. Some field members were vaccinated, others were not. But regardless of vaccination status in terms of orthopox viruses, we didn't know what other pathogens these animals might have. So that's why we're using a maximum level of protection to protect ourselves from anything that the animals might be carrying.
To clarify, back then, when people were being vaccinated who were doing field work with wildlife, were they getting the smallpox vaccine?
Yes, exactly. So people who are primarily were working with other orthopox viruses that are highly pathogenic, such as monkeypox virus, which is the causative agent for mpox, or working directly with variola virus or smallpox, The smallpox vaccine is cross-protective across other members of the genus Orthopox virus, and it's estimated to be approximately 85% efficacy across the genus. So many of us on the field team also work with other orthopox viruses, including monkeypox virus, which is the causative agent of mpox. So many of the field teams are vaccinated for that reason.
Can you share more about what your day to day experience is like when you're in the field working on an investigation?
So when we're in the field, what we generally do is we go look and we try to get traps in locations in different habitats. So we're trying to sample as many different animal species as possible. And so we want to go to forested areas, but not just generally forested areas. There might be forested areas that are spruce dominated or birch dominated or bogs and more open areas. And so we try to like sample in these diverse different types of habitats to increase the diversity of this species that we're sampling the work from. And so the workday generally consists of going out and putting traps in the field, placing them in lines or rows. We kind of do it in a systematic way. Well, there's a couple of different ways you can do it. You can do it really defined in a systematic way where you're kind of placing everything on a grid, where you can try to estimate the density of the animals within a certain area. But there's a more basic way to do it, and that's just to walk through the forest and the path of least resistance and put traps out and see what you get. It kind of depends upon the goal of your investigation, how you want to handle that. For this particular work, we didn't try to do any density estimates or that kind of thing. We would just walk out and put traps in the forest in the area where we could find them. Every trap is kind of marked with a flag and often numbered so we can go through and count and make sure we're collecting all of our traps back at the end of the day. The traps sit out overnight because most of these animals are nocturnal, so they're out in the nighttime doing their thing and trying to collect resources. In the morning, we come back and collect those traps, bring them back to a central processing area, sort of sort them out based upon the location of captures. So we're keeping them organized by different locations and habitat types. And that's where we begin the processing work. For these types of investigations, we'll basically take the trap, take the animal, put it under anesthesia. And a lot of times we'll use an inhalant anesthesia, such as isoflurane, and then that knocks the animal out. At that point, we can take it out. We can collect standard measurements, which include measuring like the total length of the animal, the tail, the hind foot, the ear, and those measurements really help us determine the species of the animal. And then we'll also collect any of the samples we need. Of course, we'll collect blood. Sometimes we can just take swabs and then release the animal alive. Other times, if we're doing a more in depth analysis, we'll actually euthanize the animal and collect internal tissues as well. For these investigations, we did euthanize these animals and we did collect spleen, kidney, liver, heart, lung, as well as some skin tissue that we can use as our diagnostic tools.
Can you explain the role of wildlife surveillance in advancing our understanding of the virus?
We ended up catching 385 animals, and there are kind of different levels of data we use to look at the infection within the animals. Just like within people, we have antibody data, we have PCR data, and we have actual detecting the viable virus particle within those samples. Antibody data indicates a previous exposure to the virus, whereas PCR positive, sort of amplifying the viral DNA, indicates a current infection. And then, of course, the gold standard for being able to confirm that a virus was present in the animal was actually identifying the virus itself. So we have these three different levels of data that we look at when we're looking at our data streams. Most of the animals, if they're positive for anything, will have antibody data, whereas a small number will have PCR positive results, and then even fewer number will have viable virus present in tissue samples.
How did historical biological specimens that had already been collected for other purposes lend strength to this work?
So when we originally first identified positive animals from Fairbanks in 2020 and 2021, we thought, well, there have to be some more evidence out here in animals within Alaska. But it's really expensive to get out in the field and do these active samplings where you're going out and play animals at the Universal Alaska Museum. He said, well, we have thousands and thousands of redback vole samples in our collection right now. Maybe we could send some to you guys and you could do some testing on those. And so we did get a tissue loan of around 200 animals from them, and we were able to identify the virus from the Denali National Park area from some samples collected in 1998 and 1999. That was, what, 17 years prior to the identification of the first human case. And so this is a really valuable resource as well to go back and do retrospective testing and to get more information. So not only in this case did we get more geographic data, we also got temporal data as far as when the animals were around at that time. We got more genetic data as well that we're able to add to our phylogenetic tree.
Has any research been done looking at like climate change that would increase rodent populations?
There have been some studies looking at climate change and seasonal factors on redback voles in general. So there were some studies out of Canada. There was actually a couple that linked berry crop production with changes within the northern redback vole community. And so they saw when it was a really bumper year of berries, they also had a bumper crop of voles. So whether there's some type of trophic cascade going on there, where some environmental conditions make it more favorable for the animals to reproduce and spread, and maybe that also might mean that the viral prevalence comes up within those populations as well. It's really hypothetical at this point in time because there's no solid data, but those are some important questions we'd like to address with some future research.
The most recent case was diagnosed on the Kenai Peninsula. Can you explain what was unique about that case?
Sure. That case first became ill during the summer of 2023. That person was immunocompromised. They were taking some drugs to fight cancer, and that caused their immune system to be weakened. And so they presented at a hospital. They said they were ill. The doctors sent them back home. And then after some time, they continued to get sick. They did have some lesions on their body, and those began to expand and get worse. So they ended up going to the hospital. And when he was back in the hospital the second time, it was noticed that this does look viral. And so there were some samples collected and sent off for whole genome sequencing. And it came back as being cowpox virus, which is a orthopox virus in it's in the same group as this virus is. And so they sent samples to CDC for further characterization. Cowpox virus is actually found across Europe. And so it would be a new finding to find that within Alaska. And upon further assessment, it was determined that this was Borealpox virus. And so, in addition to what made that unique, it's also 300 miles south of where any other human cases have been found before, since all the other cases have been identified from the Fairbanks area. So, we have this person with much more serious disease at a new location. And unfortunately, that person did end up succumbing to disease, and it was a fatal case. And that person passed away in January of 2024. But the fact that it was this new area also led us to think, well, maybe there's something else going on in the small mammal community down the Kenai Peninsula versus what we had seen in the Fairbanks area. And so there was another investigation done into the zoonotic sources. We went out and trapped animals in that region. Now, one thing I failed to mention when we were discussing some of the Fairbanks investigations was the specific species involved. The largest number of animals sampled were northern redback voles, but we also sampled quite a few squirrels. We had shrews, and there were a few other species kind of mixed in, including house mice, weasels. We tested a porcupine and a marmot. And so we had some opportunistic samples that we had collected as part of those investigations and sampled about 135 animals. And we did identify all three levels of evidence within those animals again. So we had antibody evidence, we had PCR positives, and we did able to find viable virus. And really, the one that kind of stood out as being most likely to be a regular reservoir of the virus or a species that's responsible for maintaining the virus within its population naturally was very likely the northern redback vole. We had a very high percentage of those animals be positive by antibodies, as well as positive by PCR with viral DNA. And we did get some viable virus from some of those animals as well. And then we were able to do some whole genome sequencing on those samples or from the animals and compare that to the sequences of the humans. And we actually see two clustering groups going on. We see a clustering of all the Fairbanks Borealpox virus isolates, and that includes human and animal specimens. And then we see a second clade down below that that just has the human and the animal isolate from the Kenai Peninsula area.
Multidisciplinary collaboration is foundational to One Health work. Can you explain how collaborative partnerships were formed in this investigation and how a multidisciplinary approach influenced your work?
Yeah, that was really key here to this investigation and sort of making everything happen. Bringing these teams together with people with a variety of different expertise. We had epidemiologists, we had laboratory experts, we had ecologists, veterinarians, a lot of different groups of people here collaborating to make this happen. The Alaska Division of Public Health was the group that was first notified of the event here. And so they reached out to the Poxvirus and Rabies branch at CDC in Atlanta, which is where I was working prior to coming to the Arctic Investigations Program. And then to have the local knowledge of the animals in the area, then we as a group, we reached out to the University of Alaska Fairbanks and the team at the Universal Alaska Museum of the North as far as having that local mammal knowledge to be able to go out and collect samples from these animals and have, the best results possible.
Can you explain how your role as a laboratory scientist fits in with the other disciplines involved in an epi investigation like this?
Yeah, sure. So prior to coming to Anchorage to the Arctic Investigations Program, I was in a position where I was primarily doing like disease ecology work. And really this kind of position is sort of in it's in a almost like a liaison or an intermediary between epidemiology and laboratory scientists. So our teams would go out and collect samples and bring those back to the laboratory and do the testing. And so we're sort of wearing both hats in this particular role where we're doing the field work, bringing those samples back, seeing them throughout the whole process, testing it, and then doing all the analysis afterwards. But that laboratory bench component is indispensable. It's the backbone, it's the linchpin of this whole operation because without the laboratory data to support the findings determined through just general investigation, the case is much weaker.
Can you share a bit about your career trajectory and what led you to your current role?
Sure. Yeah. I feel like for me, getting to where I am now was just sort of this series of serendipitous events. When I was first starting off as an undergrad, I was a wildlife biology major at Texas Tech University. And when I sort of wrapped up that work, I ended up getting a master's degree in biology and was focused on ecology and mammals in general. And I heard about a job opening at Colorado State University working on hantaviruses. And what they were wanting someone to do is go out in the field, trap rodents, and test those animals for hantaviruses to track the prevalence of the virus within the populations over time. And so that was really where I kind of got into the disease ecology aspect and started learning about, viral assays to detect pathogens and really linking my wildlife biology background to this public health world, which was new to me at the time. And so I worked on that for several years. And when the funding, because if all this work is dependent on funding and what's going on, the economy was kind of going bad, like let's say 2008, 2009. And so the funding was drying up. And so I ended up getting a call from someone at CDC, and they were like, "Hey, we have this project working on monkeypox virus. Would you like to come here and work on this project with us?" And so I ended up moving to Atlanta and working for the Poxvirus and Rabies Ranch for 13 years. If somebody had told me when I was an undergraduate that I'd be working for CDC, I would have been like, "Ah, no, you're crazy." But one thing led to another, and I was just able to keep my options open and explore different opportunities. And then maybe, yeah, right about a year ago is when I moved up here to Alaska and took the job at the Arctic Investigations Program.
What qualities or characteristics lead to success for a laboratory scientist?
I think having a key eye for detail, a lot of these things that you might particularly think of as laboratory scientists, doing things in the same way repeatedly over and over again to get the most consistent data, those types of things are key that I think people think of generally, but also being able to communicate with others, whether it's the epidemiologists, people in the field, other laboratorians, keeping your communication skills strong is also super important. And being able to clearly communicate what you want, what you need, as well as your findings to others is really key for advancing within the field and not just being limited to being on the bench your entire life. Not that that's a bad thing, but it's good to get out there and communicate with others as well. Yeah, so you asked about the multidisciplinary nature of this type of work and how this particular project was established. I think that that is the key to success, particularly if one particular part of the team isn't familiar with the local area or any local issues, then having that local factor is really huge. Having this multi-sectoral approach is really key to having a successful investigation. Really being sure to highlight the contributions of all team members. For me, that's really sort of the crux of the situation and really the best way to move anything forward.
Jeff, thank you so much for joining me this morning to talk to One Health students about your research and career.